Hi, welcome back to My Smart Learning. This is a lesson for Year 12s, A-level biology, and Year 13s obviously because we're starting photosynthesis. So it's useful for Year 11s who want to go into A-level biology too. So I've posted that on Show My Homework for Year 11s, 11B biology, 11Q and 11U. So let's get started. We're looking at an overview then of photosynthesis. What you know from GCSE, there's some A-level content in there too, but it's the beginning of the photosynthesis chapter and we're having a quick overview. So what we're looking at, how are plants adapted for photosynthesis? So it's a recap from year 11 when you did the photosynthesis back at GCSE, but it's also got a bit of content from the A-level spec as well when we looked at the structure of plant cells. Recall quiz, quick high five, I can't forget that. So number one, what are our three types of averages that we looked at last lesson when we looked at standard deviation? When sampling, what must you avoid? Okay, what do we need to think about? How do we do that? Number four, what is the formula for standard deviation? And the last one, what does the standard deviation tell you? Have I got those questions? Press pause. Once you're done, you can unpause and I'll go through those, right? You'll get your virtual high five at the end. So if you get these all five right. So what are the three types of averages? We've got mode, median, mean. When sampling, what must you avoid? You've got to try to avoid bias, sampling bias, and you've got to avoid chance. Number three, how do you do that? Well, to avoid sampling bias, you need to use some kind of random number generator, some random sampling technique. How do you reduce chance? You get a massive sample data. So the more data you get, you avoid chance and you have representative data. Number four, what is the formula for standard deviation? If you remember, standard deviation is the square root of, and on the top we had the sigma sign, which means the sum of, open bracket, we had x, take away x with the little bar at the top, x bar, close bracket, we squared that. So it was the data, take away the mean, we square it, then we add all of those numbers together, divided by n minus 1, n meaning the number in the data set, take away 1. So if you've got 10 things, take away 1, it becomes 9, you divide it by 9. What does the standard deviation tell you? It tells you how spread the data is from the mean. The bigger the standard deviation, the bigger the spread from the mean. The smaller the standard deviation, the less spread from the mean. The smaller number, the better, the more accurate your data set is. So let's, without further ado, what is our structure of our leaf? How are leaves adapted for photosynthesis we're looking at. So you should remember now from your GCSE, a bit of a picture here, you've got a waxy cuticle at the top, makes it waterproof, it's that shiny layer that you can sort of scrape with your fingers on a xerophytic plant. On a rubbery plant you can get a waxy layer, you can scrape it off. What's the purpose of it? It makes it waterproof. Why? Is it to stop rain from coming in? No, it's to prevent water loss. So any water is in there, water vapor, it prevents it from evaporating out. Because water is a finite resource, it's scarce, you don't want to just lose water for the sake of it, although you do from transpiration. You've got the top layer and the bottom layer, we call it the upper and lower epidermis. It's a bit like your skin on your hand, like a dermis, dermis meaning like a skin layer. You've got the upper layer and you've got the lower layer, the lower epidermis. You've also got a cuticle underneath there. What is the purpose of that skin? The transparent layer that allows light through. But I call it, it's just like my skin, it just holds everything in place. It's not very useful, just functional, sort of holds everything there. The useful bits and the tasty bits are inside. Why am I saying tasty? Because I use another analogy, it's like a sandwich. Sandwich, the bread isn't that interesting. It's what's holding the sandwich in place. It's just there so you can hold the filling, which is the most important bit, okay, that you want to eat. So obviously you got your favourite filling. I'm not going to start that debate because every time I do that in class, we digress and we don't get back to the lesson for a few minutes. So let's just say it's a cheese sandwich, cheese and tomato sandwich, or cheese and onion. So the middle bits then are the important bits. They're called the mesophyll layers. So mesophyll is just Greek, I think, or Latin for middle. It means middle layer. So you've got the two middle layers. We got the palisade mesophyll layer, named after the old shopping centre in town. No, that's a joke. You've probably forgotten because you've got short memories that the Grand Central used to be known as the Palisades. If you don't remember that, that used to be a shopping centre, but it was decrepit, it was old, it was defunct, it was broken down and it was horrible. Now they made it all fancy and now it's called Grand Central. Much nicer. We've got a nice food court. talking about food again. I'll stop. So the palisade layer is that top layer, they're long and cuboidal, and the lower layer there is the spongy mesophyll layer. Okay, so they're irregular layers there. Again, it's the mesophyll, it's the middle layer. And within there, you've got the vascular bundles, and within the vascular bundles, in these veins, you've got the xylem and you've got the phloem. Now, the sponge, if you look at the lower epidermis as well, you've got the guard cells, which open and close, and in the middle of the guard cell, you've got the stoma. Stoma is the whole. Stoma for one is singular. Plural is stomata. So they allow gas exchange. So carbon dioxide goes in, oxygen comes out, water vapor escapes, and it comes out of those stomata. The guard cells open. They become turgid during the day, so they open up because they're thicker on one side and thinner on the other side. And during the night, they become flaccid and they close up, so it prevents water loss. We did that back in the transpiration lessons, a few lessons back. So I think we did talk about this, but not into this detail. The palisade layer then is regular and cuboidal. Why? Because then they can pack together like a brick wall. So therefore, it can capture as much and absorb as much sunlight as possible. You try and capture every single photon. If there's gaps between them, you're going to lose light. Also, the vacuoles in the upper epidermis, the palisade mesophyll layer, the vacuoles push the organelles inside, pushes the cytoplasm and the organelles to the edges of the cell, so that the chloroplasts are towards the edges. So again, you don't need to penetrate very far into the cells. They can absorb as much sunlight as possible into the chlorophyll molecules inside the chloroplasts. Okay. What else have we got? The spongy layer is irregular. Why? They're irregular, so when they connect, they leave little spaces between called air spaces, and that allows the diffusion and the movement of gases for gas exchange. So that's our adaptation. The leaves are darker on the top, a bit like your skin here, and lighter on the underside. Why? Because there's more chloroplasts, more chlorophyll at the top, because that's the bit that's facing the sun. It's going to be absorbing the sunlight. The underside isn't so much. Anything that bounces off the ground and reflects back up, it'll absorb some through there. Obviously, some's penetrating straight through as well. So that's your adaptations of your leaves. Your leaves are food factories for the plant. Okay, that's where glucose is being made. So these are some of the notes I've made for you already. So we didn't mention that they've got a large surface area to volume ratio because they're flat, they're broad, and they're very, very thin. The arrangement of leaves, if you stand under a tree when it's a rainy day, or if it's a sunny day even, if you stand under a tree, if it's raining, you don't get hit by much rain because it kind of blocks like an umbrella. But also, if you look up from underneath a tree, you don't see the sky. Why? Because each of the leaves are orientated in such a way that every little bit of light, if there's a gap there and the light's hitting the ground, what a waste. You're trying to capture every single photon possible because every photon counts into producing glucose. So your leaves are arranged in a way to minimize overlapping so that they're not one on top of the other, because that's just completely blocking the light. So you want to be slightly apart, slightly that way as you go down. So you're capturing every single one. So to avoid any shadowing one over the other. They're very thin, so they can absorb as much sunlight and light can penetrate straight through them. And they're very thin to allow a short diffusion pathway. If you think about Fick's law, the thinner, the shorter the diffusion pathway, the more gas exchange you're going to get. The cuticle is waterproof, but it's also transparent. That allows light to flow through. You've got narrow upper mesophyll cells packed with chloroplast to collect sunlight. That's your palisade layer. They're like bricks, so it makes like a brick wall. And you've got lots and lots of stomata on the underside, and they can open and close during the light wave use. So therefore, at night time, you're not losing water for the sake of it. So you've got lots and lots of gas exchange. Got lots of air spaces at the bottom. In the lower mesophyll, spongy mesophyll layer, and we've got a network of xylem and phloem in the vascular bundles, so that you can transport water and minerals and glucose from the phloem down to other parts of the plant. So that's another little diagram there. Do a cross section through the leaf, and we've just looked at that. That's more of a 2D diagram compared to the previous 3D diagram. So if I zoom into that diagram then, and I look at one particular cell, in particular the palisade mesophyll layer, you can see they're packed full of chloroplast. The vacuole pushes all of that stuff to the sides so you can capture as much sunlight as possible. But if I zoom in again to a chloroplast, and coming on to sort of A-level content again, we looked at the organelles. if you look at a chloroplast, it's a double membrane, hence why you don't find them in prokaryotes, you only find them in eukaryotes. You've got double membrane outer layer. You've got like a jelly-like matrix inside. That jelly-like substance is called a stroma. Okay, it's called cytoplasm when it's inside the cell, but inside the chloroplast we call it stroma. Then you've got these little discs. Each of those little discs is called a thylakoid, and the stack of discs, one stack is called a granum. So one stack of discs is called granum. There's lots of these stacks, so the plural is grana, and then these stacks are connected by these little bridges, and these little bridges are called intergranal lamellae. Intergranal lamellae. Lamella for one bridge, lamellae with the E at the end for many bridges. So we always think about the singular and the plural. Okay, so that's the stroma there. So you've got these stacks of discs, and what's the purpose of these little discs, these thylakoids? They're the ones that are packed full of chloroplasts. There's different types of chloroplasts, but they're packed full of chloroplasts. These little proteins dotted around all over these thylakoids. So, and these little bridges, these little intergranal lamellae, they allow substances to be shared between the discs. It's like having little tower blocks, and you want to have a little bridge to go from one tower to the other so you can share food. Not literally, it's not like you're fasting, you're doing iftar or something, you're sharing food from one house to another, which you're not allowed to now because of the lockdown. You shouldn't be going seeing your friends and your family. Although some people still are in the neighbourhood, which is naughty, they shouldn't be doing that, spreading the disease and stuff. So that's what those bridges are for, intergranal lamellae. So, key structures then we talked about the thylakoid, we talked about the grana, granum for singular, intergranal lamellae, that's plural, lamella is one. Talked about stroma. The chlorophyll pigments are found in these little thylakoids, and the chloroplast is wrapped around in an envelope, a double membrane, to keep everything there. What else do we need to know from the introduction of photosynthesis? Well, the whole purpose of this is that for plants to make their own food. We can't make our own food. Okay, we're heterotrophs. We need to go and eat our food. We need to get the food, eat the food, and get the nutrients inside of us. Plants are what we know as autotrophs. Auto meaning automatic. They do it themselves. They make their own food. That's amazing. I wouldn't have to go out to Tesco once a week or Costco and risk my life to go and get some food once a week to bring it back to feed the clan. Okay, plants can just sit back, chill out, look at the sun, absorb the carbon dioxide, let it rain, happy days. They make their own food. Okay, we need to go to work, make our monies, and then go out shopping to get, or I can grow it in the garden. Obviously I could grow it in the garden, but then I'll be only eating certain vegetation and herbs. So plants though, luckily, make their own food, and then we eat the plants. They do all the hard work and we go and eat them. Okay, talking about eating is way past my lunchtime, and I need to go and have my lunch or breakfast because I haven't had breakfast either. So there's 3 stages to photosynthesis. The first stage is capturing the sunlight's energy into the chloroplast. The second stage is what we know as a light-dependent reaction, in which light energy is converted into chemical energy. That is our next lesson. That's what we're going to be coming on to next. Very complicated. This is proper A-level biochemistry. And then the third stage is also proper A-level biology, biochemistry. We're looking at the light-independent reaction. So the light-dependent depends on light. There needs to be light available. The light-independent reaction is where protons are used to reduce carbon dioxide to produce sugars and other organic molecules. That's the bit where the glucose is made. So that's what it looks like, the 3 stages. So we've got the first stage, light is captured in the chlorophyll molecules. The second stage is what we know as the light-dependent reaction, where water gets split, and the products of the water being split is used for the next stage, the third stage, which is our light-independent reaction, where we eventually get our glucose. So the stage 2, a light-dependent reaction, gives us our oxygen, byproduct, and the third stage gives us our glucose. And that is our next lesson, and then the lesson after that is that one there. So we're going to spend a whole lesson on the light-dependent reaction and a whole lesson on the light-independent reaction. And finally, our equation looks like this. Our GCSE equation looks like this, the overall word equation and similar equation for photosynthesis. Carbon dioxide plus water gives us glucose plus oxygen. The simple equation, 6 CO2s plus 6 H2Os gives us C6H12O6 plus 6 O2s. How do I remember that equation? Well, I call it the devil's equation, Y 6, 6, 6. Okay, 666, the devil's number. But it's the most important, one of the most important chemical equations or chemical reactions that are happening on Earth. Without it, we'd have no oxygen. Without it, we'd have no glucose. No oxygen, no respiration. Without it, no glucose, no food, nothing living. Okay, we're not going to have anything, we wouldn't have life as we know it. Okay, now guess what? That is GCSE. You're going on to that and that. You're going to throw that simple equation out the window now because you're going on to some hard, more technical biochemistry. Okay, and there's a lot more things to memorize and learn. But that's next time. This was just a quick overview of photosynthesis. If it's been useful and you've learned something today, I'm glad that I've been of use to you. And please keep watching us on My Smart Learning. Please like the videos, watch the videos, subscribe if you haven't done so already, and share. And I will see you next time. But before that, I can't forget to give you your virtual high five because you can't buy those in the shop. So take care. If you got all 5 questions right, I'll see you next time. Virtual high five.